During the past few weeks, we've taken a look at a lot of different things about bacteria. For example, we've taken a look at different types of molecules that different bacteria can metabolize for energy. We've also taken a look at different environments in which different types of bacteria grow best. And so if you have bacteria that just happen to grow best inside the human body, that can be a problem for us because some of those bacteria can cause disease. In other words, some of those bacteria are pathogenic. So what we'll do starting here is we'll take a look at our body's defenses against pathogenic bacteria. Recently, we've also taken a look at viruses, and you know for sure there are viruses that are pathogenic. And so when we talk about our defenses against pathogenic microorganisms, we're going to include both bacteria and viruses as examples of microorganisms that can be disease-causing. So if you consider all of the things in our body that act to defend ourselves against pathogenic microorganisms, we have a lot of stuff. We basically have a huge army of things that are on our side to protect us against pathogenic microorganisms. You can divide that big army, though, into two major branches. There is the branch that's considered to be the innate defenses, and there is the branch that's considered to be the adaptive immune defenses. The reason why things that are part of the adaptive immune defenses are considered adaptive is because these are cells that become better suited to fight off a specific pathogen the longer that it is in contact with that pathogen. So in other words, these are cells that can change, they can adapt, and become better suited at fighting off a pathogen over time. Another thing that characterizes cells that are part of the adaptive immune defense is that they produce this thing called memory. And this is the reason why it's pretty rare for someone to be infected with the same pathogen twice, is because we have cells of the adaptive immune response that produce these other types of cells called memory cells whenever they contact a particular virus, say. And so if you were to see that virus again sometime in the future, you'll have these memory cells from the first encounter that quickly recognize and quickly respond to that same virus, so that that virus can't grab a foothold right the second time around. It never is able to infect the second time around. So that you can put a face to a description, we'll give you an example of a cell that's considered to be part of the adaptive immune response. And that cell is a type of cell called a B cell or a lymphocyte. And we'll actually talk a lot more about B cells and other adaptive immune defenders later on. So we'll just leave it kind of general for now that a B cell is an example of a cell that's adaptive because it can do these two things that we just listed. If you consider the other branch of our army that defends us against microorganisms, the innate defenses, what makes them different is that, well, they're not adaptive, right? So in other words, these are components that have ways of fighting off pathogens, they have ways of defending us against pathogens, but they don't really become better suited to a particular pathogen over time, right? They just kind of keep doing the same old thing over and over again to help protect us against pathogens. So in other words, these are, if you consider the innate immune defenses, these are components whose protective functions are set and programmed in at birth. They don't really become more adaptive, they don't really change over time. So examples of things that are considered to be part of our innate defenses include the skin, mucous membranes, and different types of cells. For example, this type of cell called a neutrophil. And so we'll actually say more about what each one of these things does to protect us against pathogens. But this is basically what our army is, our army that protects us against pathogens. We'll actually save the discussion for adaptive immune defenses next week. So we'll say a lot about that next week. What we'll do in this video is focus just on these guys, the innate defenses, and we'll talk about what all these different things can do to protect us against pathogens. So if you consider then just the innate defenses against pathogens, you can actually divide that up into two branches. There's the part that's considered to be our first line of defense against pathogens, and then you have things that are that are found in the blood and the lymph, right? So you have our first line defense, and if for some reason our first line of defense is breached, that's okay, right? Because we have innate immune defenses that are also found inside in the blood and the lymph. If you're considering our first line defenders, that's basically two things. It's the skin and the mucous membranes. And when you're talking about mucous membranes, you find these things lining different tracts in the body. So for example, there are mucous membranes that line the respiratory tract. There are some that line the gastrointestinal tract, and there are mucous membranes that line the genitourinary tract. If you're considering the components, the innate components of the blood and the lymph, that's basically comprised of two different things, right? So there are innate cells, innate immune cells that are found in the blood and the lymph, and you have a type of protein called complement proteins that are found in the blood and the lymph that can protect us against microbes. And so again, this is a general outline of what the innate immune defenses are made up of. We'll actually end up saying more about what each one of these things can do to protect us against microbes. Where we'll start though is right here. So we'll first take a look at our first line defenders and see what all they can do to protect us against microbes. So again, when you're considering the first line of defense, what you're talking about is the skin and also the mucous membranes. And what we'll do here is we'll describe some things that both of these first line defenders can do to protect us against pathogens. So one of the things that they do is that they act as just a physical impediment or a physical barrier that makes it more difficult for pathogens to gain access to the inside of our body. And the reason why both the skin and the mucous membranes are good physical barriers is because if you consider the cells that make up the skin and the cells that make up the mucous membranes, they're attached to one another in a relatively stable, tight way. So it just makes it more difficult then for that lining of cells to be penetrated by things like bacteria. So they provide a physical barrier as a way of defense. Another thing that the skin and the mucous membranes do to defend us against bacteria is that they house other bacteria, right? So they fight fire with fire. So they prevent pathogenic bacteria from growing by housing friendly bacteria or normal microbiota. And so if you remember from the first week of class, we talked about normal microbiota as the collection of bacteria that normally live in or on the body and are not harmful to the body, and in fact a lot of them do really helpful things. And so the way that normal microbiota that you find on the skin and mucous membranes, the way that they help protect us against pathogens, well, they do two things. So one of the things they do is that they just take up a lot of space, and they eat up all of the nutrients so that there is less space and there are less nutrients for pathogenic bacteria to use. So they just physically take up all of that stuff. Some other normal microbiota actually release substances that kill off other bacteria, including some pathogens, right? So these are, again, both useful things that our normal microbiota do to protect us against pathogens. One other thing that the skin and the mucous membranes can do to protect us against pathogens is that they can actually themselves release substances that are antimicrobial, that can kill off pathogens. And so these are three examples then of antimicrobial substances that are secreted by the skin and or the mucous membranes. So it's lysozyme, peroxidase, and lactoferrin. Where you find lysozyme is in a few places. So you find it in tears and saliva and also in phagocytes, right? So phagocytes isn't really skin or mucous membranes, but we're including it here. So we're just giving you a heads up that you also find this stuff inside phagocytes. In other words, cells that are able to engulf and eat other things, other pieces of debris. Specifically, what lysozyme does is that it breaks up, it cleaves some of the connections that you find holding together peptidoglycan that is found in the cell wall of bacteria, if you remember. Right, so this is one of our defenses against infection. Another thing that is secreted by the skin and the mucous membranes that defends us against pathogens is this enzyme called peroxidase. You find these in specific places, including the saliva, phagocytes, and actually also in milk. Whenever you see this suffix ase at the end of the word, then it tells you that it's a type of enzyme. And specifically what this enzyme does is it catalyzes this reaction. It turns hydrogen peroxide, which is one of the byproducts of aerobic respiration, into these things called oxygen radicals. And oxygen radicals, it turns out, they can really mess cells up, right? Because they react to all kinds of different things to change up the makeup of a cell. It turns out that these things can harm our own cells, but this peroxidase is confined so that hopefully it's only harming bacterial cells or cells that are trying to cause an infection. Right? So this is another defense, another innate defense that we have that's secreted by the skin and the mucous membranes. This last thing this last antimicrobial that's secreted by skin and mucous membranes is this stuff called lactoferrin. Basically what lactoferrin is, it's an iron sponge. Right? So you might see this term ferrin or fer, and you know that refers to iron. And so lactoferrin is this stuff that attaches to any free-floating iron that's in a particular environment for the purpose of removing that iron from the environment. And the reason why that is a useful antimicrobial substance is because bacteria actually need iron. So bacteria require iron because that iron is needed to make some of the parts of the electron transport chain. So if you have this lactoferrin that's basically robbing the bacteria of any iron in the environment, then it prevents those bacteria from growing and dividing. So specifically where you find lactoferrin, it's found in the same place that peroxidase is found, in the saliva, the phagocytes, and also the milk. And again, here's in words what lactoferrin does. It removes free iron from the environment so that pathogens like bacteria aren't able to use it. So again, this is all stuff that both the skin and the mucous membranes can do to protect us against pathogens. If you consider, though, just the mucous membranes, they can do some additional things to protect us against pathogens. And specifically what they can do is that there are portions of different tracts in the body that can actually move microbes towards the exterior of the body so that hopefully those microbes can get expelled. Right? So there are some mucous membranes that can physically expel microbes from the body. An example of where you find this is in the respiratory tract. So if you take a look at portions of the respiratory tract, like inside the lungs, there are these hair-like extensions that move out away from the cells that line that tract, called cilia. And the cilia can move, right? They can beat back and forth. And the purpose of that, or one of the purposes of that, is so that they can move along any debris or bacteria that might have become lodged in the lungs out towards the throat, where they can hopefully be coughed out. So this whole mechanism of moving along any debris or bacteria in the respiratory tract towards the upper part of the tract where they can hopefully get expelled, that's called the ciliary escalator. And again, it's another defense that is provided by the mucous membranes. So if we take you back to this chart, kind of outlining the parts of the innate immune defenses, we've just taken a look at what these things can do, what these first-line defenders can do to protect us against microorganisms. What we'll do next is switch over to the other branch of the innate defenses, those parts that are found in the blood and the lymph, and take a look at what they can do. So first we'll take a look at cells that are found in the blood and the lymph, more specifically innate cells found in the blood and the lymph, and it turns out that these cells can do one of three things to protect us against pathogens. So it turns out that there are some innate immune cells that can do phagocytosis. And so you might remember from cell biology that what phagocytosis is, it's the process of one cell eating another cell, or one cell eating debris. So phagocytosis is the engulfing of solid particles. For example, we have innate immune cells that can actually engulf entire bacteria to hopefully kill off those bacteria. Right? So that's just one thing that the innate immune cells in the blood can do. Another useful thing that innate immune cells can do, well, there are some innate immune cells that can induce inflammation. And basically what inflammation is, it's the rush of blood fluids, which contain white blood cells and proteins, to a localized area. And so the reason why this thing would be useful, the reason why it would be useful to have blood fluids rush towards a particular area, is because if that area happens to contain pathogens, then those blood fluids will contain these white blood cells that will fight off those pathogens. So that's a useful thing for us. And then a third general thing that innate immune cells can do to protect us against pathogens is that there are some that can induce fever. And basically what fever is that an increase in body temperature that's done to protect us against infection. And so again, these are all things that innate immune cells in our blood and lymph can do to protect us against infection. One thing that would be useful is if these cells knew to do these things only if it ran across something bad. In other words, only if it ran across a bacterium or a virus that is looking to cause disease. Right? You wouldn't want these innate cells to start doing all of these things any old time, or you wouldn't want these cells to start doing these things when they come across cells that are normally found in the body, because these things could actually end up damaging our own cells. So somehow these innate immune cells know to engage in these activities only if it runs across a foreign thing, a foreign bacterium or a virus. And what we'll do next is we'll explain, well, how do these cells know to do this only when they run across something that is foreign and potentially infectious? And the key are these receptors that are found actually on and in these innate immune cells. Right? So if you consider this thing, so if this thing represents one of our innate immune cells, it turns out that all across the surface of this cell are these receptors. And what these receptors can recognize and what they can attach to are molecules that tend to be found only on bacteria. And so it's only when these receptors are engaged, it's only when they bind to something, does the cell become activated. And because, again, these receptors only can recognize things that are only found on bacteria, that's why this cell only responds typically to foreign things like bacteria and also viruses. It turns out that these receptors are found not only on the outside of our innate immune cells, but they are also found inside our innate immune cells in the cytoplasm of our innate immune cells. And so you're probably looking at that and thinking, well, that's kind of dumb. What use would that be? How would these receptors ever bind to something if they're stuck inside that cell? Well, keep in mind that there are actually some pathogens out there that purposefully burrow their way in or make their way in to some of our cells as part of their infection process. And so whenever you have that happen, whenever you have a pathogen make its way inside of our own cells, one of the ways that our cells know, one of the ways our cells knows to respond to that pathogen is by having receptors inside of it, right? So if a bacteria makes its way inside of a cell, there's a good chance that it'll run into this receptor that's inside of our cell. Because that receptor recognizes things that are found on bacteria, that would then call this cell into action to do stuff to hopefully kill or expel that bacteria. Right? So our innate immune cells, again, have receptors that allow them to recognize things that are found on bacteria. Lets them differentiate between bacteria and cells that are normally found in our body. What these receptors are called is they're called pattern recognition receptors, or PRRs. And again, to sum up everything we've just said, what these PRRs do is that they recognize and bind to large molecular patterns that are found only on microbes. So examples of molecules that are found only on microbes include things like flagellin, which is the protein that makes up flagella, lipopolysaccharide, right? Our cells don't have LPS, but bacteria have LPS. Teichoic acids are only found on bacteria. Lipoproteins are found on bacteria, but not our own cells. And you have viral RNA. It's a type of nucleic acid that tend to be tends to be found in viruses, but not in our own cells. Another thing to know about these pattern recognition receptors is that, well, it turns out that they're not only found on innate immune cells, but they're found on other cells in the body as well. And actually they're found on cells that may or may not have anything to do with protecting us against pathogens. And so we'll say a little bit more about that later on in the quarter. But for now, we'll just kind of plant this seed that these pattern recognition receptors are found not only on innate cells, not only in immune cells, but also other cells of the body. All right, so we have these cells in our blood that can protect us against pathogens. The way that they know what to attack is because they have these things called pattern recognition receptors. What we'll do next is we'll say a few things about this other thing that you find in the blood and the lymph that can also act to protect us against pathogens, and that's the complement proteins. And it turns out that these complement proteins, they're made by liver cells. Turns out that other cells make them too, but liver cells make a lot of the complement proteins that you find in the blood and also the lymph. The reason why these things are called complement proteins is it's not because they say nice things all the time, but it's because they complement the activity of something else. In other words, they work together with another immune component in order to get rid of bacteria. So when these proteins were first discovered, what they were found to be doing was they were working together with, or they were complementing the activity of these molecules called antibodies in order to get rid of bacteria. So that's why these proteins are called complement proteins. We actually know now that they do a lot more stuff other than just working together with antibodies, and we'll talk more about that actually in class. But I'm not actually right now. We'll mention some things that complement proteins can do. So one of the things that complement proteins can do is that they can coat the surface of any invading bacterial cells, and what that does is that it signals phagocytes to come eat it. Right? So it turns out that bacteria can be eaten or phagocytized even if there aren't any complement proteins coating them. But if there are complement proteins that are coating the bacterial surface, it just makes it more likely that that bacterium will be phagocytized or eaten. Right? So that's one thing that complement proteins can do. Another thing that complement proteins can do is that they can actually bind to some of our own cells and cause inflammation and induce inflammation. And we talked earlier about why inflammation might be useful if there's an infection occurring. Right? So complement proteins can encourage that process. And then a third thing that complement proteins can do is that they can actually directly attack bacterial cells. They can basically poke holes in the membrane of bacterial cells. And so we'll talk about how that happens in class. For now, we'll end this little part on complement proteins by sharing a term. That term is the complement system. And so whenever you hear somebody talking about the complement system, basically what that is, it's the collection of all the different complement proteins found in the body and all the different things that complement proteins can do. So you'll actually see in class that there are many different types of complement proteins. Each different type of complement protein does a different thing. And so the collection of all of those proteins and all of those activities that complement proteins do, collectively, it's called the complement system. Okay, so what we'll do now is zoom back out to take a look at the big picture. So again, this is kind of an overview of all the different parts of the innate immune defenses. We've already said some stuff about these guys. We've said some really quick things about these guys over here. What we'll do next is say more. We'll focus just on the innate cells of the immune system and take a look at what those cells are and what those cells can do. So we'll take a look at that next. So if you consider not just the innate cells found in the blood, but all of the cells found in the blood, we'll start there. You basically can divide that into three different categories. So there are cells in the blood that are called red blood cells. Another term for that is erythrocytes. And basically what red blood cells do is that these are the guys that carry oxygen to different parts of the body. And oxygen is needed for Aerobic respiration, right? It's needed for as a final electron acceptor at the end of the electron transport chain. So that's one major type of cell that's found in the blood. Another major type of cell found in the blood are these cells called megakaryocytes. And the reason why they are important is because they produce these things called platelets. And what platelets do is that they encourage clotting. They make clotting possible. And clotting is important because it stops bleeding, so it prevents us from losing all of our blood if we're ever injured. A third major type of cell that you find in the blood are white blood cells. Another term for white blood cell is leukocyte. And so it turns out that this is the type of cell in the blood that we're going to be focusing in on here. It turns out, we'll mention here that what white blood cells do, their basic function is that they help fight off infections. If you consider just white blood cells, there are 4 major types of white blood cells, or at least we'll consider there to be 4 major types of white blood cells for this video. So there are white blood cells that are categorized, or there's white blood cells that are called macrophages. There are some that are called dendritic cells. There's a population that are called granulocytes, and there's a type of white blood cell called lymphocytes. And so what we'll do is go through each one of these guys, these white blood cells, and mention what they all do. So first, if you consider macrophages, it turns out that macrophages aren't found in the blood. Right? So it turns out that macrophages are cells that have migrated out of the blood and are now housed in the body's tissues. And it turns out that you'll see in your textbook, for example, that different types of macrophages are named according to which tissue they're found in. Right? So if you, for example, are talking about macrophages that are found in the liver tissue, those are called Kupffer cells. If you are talking about macrophages that's found in nervous tissue, those are called microglial cells. Macrophages found in the lungs are called alveolar macrophages. So we're just kind of highlighting the point that macrophages are found in different tissues of the body. They can be called different things depending on which specific tissue they're found in. What macrophages do is that they eat stuff, right? So macrophages are an example of a cell that is, or that has phagocytic activity. Not only that, but macrophages can be activated to be able to do other things, right? So it's kind of like a video game. There's video games where you're driving a car that's able to shoot stuff, but if you pick up a special little gem, you're able to shoot other stuff too. So the same thing with the macrophage. It's kind of by default has this weapon against bacteria. It can eat them. But if it's activated by another type of white blood cell, which we'll talk about later, it can also do something else, which is to release toxic substances that can kill bacteria and other types of microbes. Right, so that's the story on macrophages. We'll actually save the story on dendritic cells. We'll tell that story later on when we take a look at adaptive immunity next week. If you consider lymphocytes, we'll actually save lymphocytes, the more detailed discussion on lymphocytes later, but we'll tell you now that examples of white blood cells that are considered to be lymphocytes include these things called B cells. T cells are another example of lymphocytes, and there are these things called natural killer cells that are examples of lymphocytes. Right? If you consider the granulocytes, there are 3 major types of granulocytes, and those are these things called neutrophils, these things called basophils, and these things called eosinophils. And so you might be able to tell from the pictures here, the reason why these cells are called granulocytes is because if you stain them a certain way and you look at them under the microscope, they contain these little granules inside. And so the granules contain different things depending on whether you're talking about a neutrophil, a basophil, or an eosinophil. So what we'll do here is that we'll take a look at, we'll explain what these different types of granulocytes can do. First of all, if you consider a neutrophil, it turns out that there are 2 names for this type of cell. So not only are they called neutrophils, but also in your book you might see to them being referred to as polymorphonuclear cells, or PMNs. If you break down this term, polymorphonuclear, literally it means many-shaped nucleus, which is appropriate because if you take a look at these cells under the microscope and you focus in on their nucleus, it looks like it's made up of different shapes kind of strung together. So that's why it's called a polymorphonuclear cell, or a PMN. What these neutrophils or PMNs can do, their major antimicrobial activities that they can eat bacteria. So it's a phagocyte, kind of like a macrophage is also a phagocyte. Not only that, but they can also release digestive enzymes and antimicrobial substances that can kill off bacteria. And just one more thing about neutrophils, they happen to be, if you consider all three of these types of granulocytes, neutrophils are the most abundant out of the three. So you find a lot of neutrophils in comparison to basophils and eosinophils in the blood. If you consider this granulocyte, the basophil, what it does is that it's primarily involved in inflammation. So the granules inside of basophils contain this substance called histamine, and when histamine is released, it actually causes, it helps inflammation to occur. And so we'll say a little bit more about that later on. And actually, before we say some stuff about this last granulocyte, the eosinophil, how about if we rewind and mention that if you consider the little granules that you find in neutrophils, what they contain are these things. They contain these digestive enzymes and antimicrobial substances that are used to kill off bacteria. Right, so that's what those granules contain. These granules contain histamine. What eosinophils do is that they have antimicrobial substances too. So if you take a look at their little granules, they also contain things that can kill off bacteria. And actually, more so than bacteria, what these eosinophils seem to be especially equipped at fighting against are parasitic worms. So people don't know exactly what it is about eosinophils that make them especially good against worms, but they are. And one other thing about eosinophils, one other thing that they do is that they actually contain substances that can reduce inflammation. And reducing inflammation is a useful thing, because inflammation that's allowed to happen over a long period of time can actually damage our own cells. Right, so again, these are three types of granulocytes. Granulocytes are a type of white blood cell. We'll actually toss one more type of white blood cell, or leukocyte, into the mix. And it's a type of cell called a mast cell. And we'll put it right here, right next to the basophil. So I'm not drawing an arrow to it from granulocyte because technically mast cells are not considered to be a type of granulocyte. But the reason why I'm placing it here on this chart is because mast cells do something, well, their function is very similar to what basophils do. So if you take a look inside mast cells, they also contain histamines that they can release and induce inflammation. So that's why I say they have basically the same function as these basophils. There's a difference though, right? So if you consider basophils, where they are found, it's mostly in the blood. On the other hand, if you consider where mast cells are found, they're not found in the blood, but instead mostly they're found in the surrounding tissue. So they're similar in that they do similar things, in that they release histamine, but they're found in different places. So these are a lot of white blood cells here that do different things to protect us against infection. If you consider dendritic cells and lymphocytes, those are white blood cells that are considered part of the adaptive immune response. So we'll wait to talk about those cells later, next week. What we'll focus in on here are cells that are considered to be part of the innate defenses. And so those would be these guys, the macrophages, and also the granulocytes, and the mast cells as well. So we'll say more about these cells right now. But before we take a look at the details of what each one of these cells can do, we'll mention some general things first. And one of the things to mention about these cells is that, well, we've already mentioned that they have a way of identifying foreign cells like bacteria and knowing to act only against those cells and not to act against the cells in our own body. The reason why that is because if you take a look closely at all of these cells, they contain these things called pattern recognition receptors, which we've already talked about. What we'll mention here is that there are actually different types of pattern recognition receptors that recognize different things and cause different things to happen after something attaches to them. So we'll list those things here. So we'll list those types here. Again, 3 types of pattern recognition receptors. There are these things called toll-like receptors, represented by these green things. So toll-like receptors are an example of a pattern recognition receptor. There are these things called nod-like receptors that are another example of a pattern recognition receptor. And there are these things called rig-like receptors that are yet another example of a pattern recognition receptor. If you consider these toll-like receptors, here's the skinny on the toll-like receptors. First of all, where you find toll-like receptors is on the surface of macrophages. So they're embedded within the membrane of our macrophages. It turns out, though, that macrophages aren't the only type of cell that contain toll-like receptors. Other cells have them too. So cells like B cells and dendritic cells and even other non-immune cells contain them. But we're focusing in on the innate immune response, so that's why we have macrophages kind of on top here, because those are an example of innate immune cells. Another thing about toll-like receptors is that, well, it turns out that there are different types of toll-like receptors. And what makes each type of toll-like receptor different from one another is what they recognize, right? So all toll-like receptors recognize some general molecular pattern that tends to be found only on bacteria. But some toll-like receptors recognize different general molecular patterns as compared to others. Right? So for example, you have some toll-like receptors that tend to recognize stuff that you find on flagella. There are other types of toll-like receptors that recognize lipopolysaccharide. Right? So there are different types of toll-like receptors, again, that recognize different foreign molecular patterns. If you consider these nod-like receptors, well, you can see based off of the picture here that where you find them is inside, in the cytoplasm of cells. Specifically, you find them in the cytoplasm of macrophages. And what happens, so when these nod-like receptors end up binding to a foreign cell, like a bacterium, it causes that macrophage to do a specific thing. And what that is that it causes that macrophage to release messenger molecules that end up causing inflammation. Right? So these nod-like receptors are associated with inflammation. And if you consider this last type of pattern recognition receptor called a rig-like receptor, again, you can tell from the picture where you find these. They are also found in the cytoplasm of cells. And what they are really good at recognizing are things that you find typically on viruses. So they recognize general molecular pattern found on viruses. So these are one of the things that would cause a cell to put up its defenses if it's infected then with the virus. Right? So again, what we're taking a look at here are cells that are in the blood that are innate, that are part of our innate defenses. They know how to tell good guys apart from the bad guys because of these things called pattern recognition receptors. These are just 3 examples of pattern recognition receptors. Another general thing we'll mention about these innate cells of the blood before we get into more detail as far as what they can do is this. Well, it turns out that a lot of these cells coordinate their activities with one another. And so if you're going to coordinate activities with a different type of cell, you're going to have a way of communicating with that cell. And so it turns out that the cells of the immune system, so leukocytes, they use a specific molecule to communicate with one another. And so what that molecule is called, well, they're called cytokines. So leukocytes communicate with each other through these molecules called cytokines. And what cytokines are, again, is that they are signaling molecules that are released by leukocytes and recognized by other leukocytes. And so again, this is the way that white blood cells or leukocytes talk to one another. So if you were to see it in pictures, white blood cells talk to one another by releasing different types of cytokines that can be bound to and recognized by other white blood cells that end up then responding to that cytokine in a specific way. It turns out that there are different classes of cytokines, and each class of cytokine causes white blood cells to do different things. And so we'll list those different classes of cytokines right now. So here they are. So these are all different classes of cytokines again. There are these things called chemokines, these things called colony stimulating factor, these things called interferons, tumor necrosis factor, and interleukins. The reason why this type of cytokine is called a chemokine is because they cause chemotaxis to occur. And so you might remember that what chemotaxis is, it's the attraction of cells towards a certain area. Right? So again, chemokines, they're caused that because they're a type of cytokine that causes chemotaxis, or attraction of white blood cells to a specific area. The reason why this class of cytokines are called colony stimulating factor is because they induce cell division. Specifically, they cause cell division in white blood cells, and after those cells divide, oftentimes they cause them to differentiate or become other types of white blood cells. If you consider this class of cytokine, the reason why they're called interferons is because they interfere with the division, the replication of viruses. So these are cytokines that are really good at causing our cells to defend themselves against viral infections. Tumor necrosis factor, you might be able to tell from the name that the reason why this type of cytokine is called this is because when it was first discovered, it was found to do things that resulted in the death of cancer cells or tumors. And so one of the ways of inducing death is by way of this thing called apoptosis or apoptosis. And basically what apoptosis is programmed cell death. So tumor necrosis factor is a type of cytokine that induces that programmed cell death, which is useful in certain situations. It's useful to kill off certain types of white blood cells after they've performed their function, for example. Not only that, but tumor necrosis factor is also involved in inducing an inflammatory response. And again, we've already mentioned why inflammation might be a useful thing if there's an infection occurring. This last class of cytokine, called interleukins, they don't really do one specific thing. So these are cytokines that actually do things all across the board. They have a very varied function. They have many different roles. So for example, there are some interleukins that cause fever. There's some that cause inflammation. There's some that activate certain white blood cells. So they're really varied. So again, these are all different types of cytokines. Cytokines are just the signaling molecules that white blood cells use to talk to one another. Taking it back to the big picture, so again we're taking a look at the innate cells that are found in the blood and the lymph. We've already talked about how these cells can tell the difference between good things and bad things, so how they can tell the difference between pathogens and the cells of our own body. And we've also talked about how these cells communicate with one another. So now that all that groundwork has been laid, what we'll do next is actually talk about what these cells can do to eliminate pathogens. So one of the major things that these cells can do, we mentioned way in the beginning, is that there are some of these cells that are phagocytic. In other words, they can eat and digest bacteria, for example. Another major thing that these cells can do is that some of them can induce inflammation. And we mentioned why inflammation might be a useful thing if there's an infection. So what we'll do next, for the rest of this video, is we'll talk about these things. So we'll talk about what occurs during phagocytosis, we'll talk about what occurs during inflammation, and then towards the very end we'll actually talk about what fever is all about and why that might be useful during an infection. So next we'll take a look at how things like macrophages and neutrophils can phagocytize foreign debris, or even foreign bacteria, which are basically big pieces of debris. And so in order for one of our phagocytes to phagocytize a bacterium, for example, some things have to happen. So first of all, we'll point out some components of a phagocyte that play a big role in helping it to eat a bacterium. So one of the important things is this organelle that you find inside of phagocytes and other cells as well, called a lysosome. And the reason why it's called a lysosome is because it contains enzymes that can lyse things, that can break things apart. And so that's what these orange things represent. Those are the hydrolytic enzymes that can break stuff apart. On the surface of a phagocyte are several different molecules, several different receptors, and so we're highlighting just 3 of those types of molecules right here. So it turns out that phagocytes, like macrophages and neutrophils, contain a pattern recognition receptor, specifically a type of pattern recognition receptor called a toll-like receptor. And you'll remember that what this allows the phagocyte to do is to be able to recognize things that you find only on foreign cells. So this is what allows the phagocyte to just attack bacteria and hopefully leave our own cells alone. Another receptor, another molecule that you find on the surface of phagocytes is represented by this thing. This is a receptor for complement proteins. So phagocytes are able to bind to complement proteins. We'll see why that's useful here in a second. This last receptor that we're highlighting here, this orange thing, is a receptor called an Fc receptor. Here's why it's called an Fc receptor, because it binds to a portion of antibodies called the Fc region. Right? So here's what that means. If you consider an antibody molecule, generally antibody molecules have this capital Y shape. This portion of the antibody molecule down here, so the long stem portion of the Y, is called the Fc portion of the antibody. And so what we're saying is that phagocytes have a receptor on their surface that actually attaches to this portion of antibodies. So that's why it's called an Fc receptor, because it binds to the Fc portion of antibody molecules. And again, we'll see why it would be useful to have an Fc receptor here in a second. Okay, so then in order for this phagocyte to phagocytize this foreign bacterium, the first thing that has to happen is that it has to get close to that bacterium and then attach to that bacterium. And so remember that the term used to describe that process is chemotaxis. So again, chemotaxis is the attraction of a cell, like a phagocyte, to a certain area, like an area that contains bacteria. It turns out that there are different things that could cause chemotaxis of phagocytes. In other words, there are different things that can cause phagocytes to rush to a particular area. For example, bacterial parts like lipopolysaccharide and teichoic acid. So if there are any of these things around, that's one thing that would attract phagocytes to a particular area. Makes sense, right? Because phagocytes have these toll-like receptors that can actually bind to things like lipopolysaccharide and teichoic acids. Another thing that would cause phagocytes to rush towards a particular area are certain cytokines. So we just finished talking about a type of cytokine called a chemokine that acts to attract white blood cells towards a particular area. Right? So if there's another white blood cell in the area that has maybe already encountered that bacterium, that would cause that white blood cell then to start spewing out these chemokines that act to help attract reinforcement, right? To help attract phagocytes into the area that might hopefully eat up that bacterial cell. And one last thing that can cause phagocytes to rush towards a particular area are complement proteins. Right? So there are some complement proteins that can cause chemotaxis. So let's say then that this phagocyte ends up rushing towards this area where the bacteria are for the purpose of hopefully getting rid of that bacteria. The reason why that phagocyte can bind to that bacterial cell, again, is because it has this toll-like receptor that can recognize and bind to molecules that you only find on bacteria. Right? So that's the reason why the phagocyte can attach to it. It turns out that there are things that would make it more likely that this phagocyte would attach to this bacterial cell. So here's an example of something that would make it more likely for this attaching to occur. So first of all, the take-home point here in this little part is that toll-like receptors, again, they allow the phagocyte to bind to the microbe. If, however, that microbe happens to be coated with particular types of proteins, for example, complement proteins that are made by our liver and other cells, It turns out that if there's complement coating the surface of a bacterial cell, that makes it more likely that this phagocyte would be able to bind to it. Basically because now you have two reasons for this phagocyte to bind to this bacterial cell. Not only are there things like lipopolysaccharides, teichoic acids, maybe flagellin on that bacterium that would be recognized by the toll-like receptor, but now there's also complement proteins on the bacterium. Remember that phagocytes have a receptor for complement proteins, right? So now there are two receptors that could possibly engage that bacterial cell. Here's another thing that would increase the chance that a phagocyte would bind to a bacterial cell. And that's if that bacterial cell happens to be coated with antibodies that are produced by other types of white blood cells. The reason why having antibodies on a bacterium would make it more likely that it would be bound by a phagocyte is because, again, there are now two reasons for this phagocyte to bind to that bacterial cell. One, it has a toll-like receptor that can recognize things on that bacterial cell. Another is that phagocytes have this Fc receptor. Remember what that Fc receptor does is that it binds to that long stem portion of the antibody molecule. Right, so again, here you have, if you're the phagocyte, two reasons to bind to that bacterial cell. So the point here is that phagocytes, even if there aren't any complement proteins and even if there aren't any antibodies bound to that bacterial cell, that phagocyte could still potentially attach to it because of that toll-like receptor. But if there happens to be complement proteins, and if there happens to be antibodies binding to it, that just increases the chance that the phagocyte will bind to that bacterial cell. There's a term used to describe this, right? So complement proteins and antibodies, those are both examples of something called an opsonin. And whenever you're talking about an opsonin, what that is, it's a protein that can bind to a foreign particle or even a bacterium and make it more likely for that particle or bacterium to be phagocytized. And a term that is associated with opsonin, or basically just a spin on opsonin, is this term opsonization. Right? And what opsonization is then, it's basically described the process that, it describes the thing that opsonins do. Right? So opsonization is making a foreign particle more likely to be phagocytized by coating it with a protein, specifically either a complement protein or complement proteins or antibodies, which are also a type of protein. So that's how a phagocyte ends up binding to a bacterial cell. After it's bound, it then allows that phagocyte to engulf, to take in that bacterial cell, kind of like this. After it's inside that bacterial cell, you see how, after it's inside the phagocyte, you see that this bacterial cell now is enclosed in its own little membrane. And so what this entire structure is called, it's called a phagosome. So again, a phagosome is a foreign particle that has been, so that should say engulfed by a phagocyte, and then after it's engulfed, it ends up being enclosed in its own membrane that's formed by the outer membrane of the phagocyte just getting pinched off after it becomes engulfed. So the next few steps during phagocytosis is all about then trying to digest or to kill that bacterial cell. So one of the things that happens in order to do that is that there are proton pumps actually in the membrane of the phagosome that ends up bringing in a whole bunch of protons. So in other words, the acidity inside the phagosome ends up increasing. So this increased acidity inside of the phagosome actually ends up killing some types of bacteria. But if that does not kill the bacteria, here's what does. So it turns out that the major reason that protons are pumped inside of the phagosome is because it prepares the way for these lysozyme enzymes, for these hydrolytic enzymes inside of the lysosome to work. Right? So these are the things that's going to ultimately do the most damage to that bacterial cell. It turns out that these enzymes inside the lysosome work best in acidic conditions. Right? So all of this proton pumping inside of the phagosome is just to pave the way for the big guns to be able to work. So here's what we mean. So if you were able to see what happens next, it turns out that this lysosome ends up fusing together with the phagosome for the purpose of giving these enzymes access to the bacterium so that these enzymes can break down that bacteria. So whenever you have this structure that just formed, right, the phagosome that fused together with the lysosome, what this entire thing is called is the phagolysosome. So again, phagolysosome is the phagosome that has become fused together with the lysosome. The reason why that fusion happens is to allow these enzymes to start breaking down that bacterial cell, kind of like this. But before we show you that bacterial cell getting broken down, it turns out that there are other things that happen inside this phagolysosome that act to try to kill that bacterium. So not only do you have an acidic environment, not only do you have these hydrolytic enzymes that are now primed to do their job, but also there are these enzymes inside of the phagolysosome that end up producing oxygen radicals. And another term for an oxygen radical is reactive oxygen species, so that's what ROS stands for. So basically, if you're this bacterial cell, you are just about to have a really, really bad day, because you have three things working against you, trying to kill you, right? You have an acidic environment, you have these hydrolytic enzymes, and you have these toxic reactive oxygen species that all together hopefully end up breaking down, killing, digesting that bacterial cell. So what ends up occurring next is basically all of that debris, all of those bacterial bits and pieces end up getting expelled from the phagocyte. It turns out though that if the type of phagocyte you're talking about is a macrophage, something additional happens, and we'll show you that next. So all of this stuff again ends up getting expelled. But if you are considering a macrophage, what you find in the phagolysosome of macrophages is a type of protein. And what that protein does is that it actually binds to some of these bacterial fragments that ended up just being produced. And so what ends up occurring next is that in all phagocytes, all of this bacterial debris ends up being expelled. But if it happens to be a phagocyte that is a macrophage, right, so all of that stuff is expelled all right, but some of it ends up getting left behind because it ends up getting loaded onto this protein, and that protein ends up being embedded in the membrane. Next week we'll talk more about why this is useful, but for now we'll just quickly mention that basically what this macrophage is doing is that it's showing off a piece of that bacterium so that it can alert another type of white blood cell to get called into action. Right? So it's a way of showing off or presenting a piece of a bacterium to a white blood cell to tell that white blood cell, hey, something bad is happening, you need to get activated as a result of that. Right? Then to sum that up, what happens towards the end of this process of phagocytosis is that all the digested fragments, all the cut up bits and pieces of bacteria end up getting expelled. If this phagocyte that we're talking about happens to be a macrophage, then some of those fragments end up remaining in the membrane, kind of like we're showing you here. Again, the reason for doing that is because this macrophage is presenting bits and pieces of the macrophage, or of the bacterium, to other types of white blood cells. Right? So again, it's only macrophages that do this. If it's a neutrophil that's doing phagocytosis, what it does at the end is that it just expels the stuff, but it doesn't do this presenting of any of the pieces. It's kind of like a trophy, right? It's showing off what it just did. Neutrophils don't show off bacterial pieces like trophies. It's just macrophages that do that. Right? So that's then what happens during phagocytosis. We're talking about phagocytosis because we're talking about things that innate cells of the blood can do to help get rid of pathogens. And it turns out that macrophages and neutrophils are both cells that can do phagocytosis. What we'll do next is we'll take a look at this other thing that other innate cells of the blood can do to get rid of pathogens, and that's inflammation. And so again, you have basophils and mast cells that are examples of cells that can help to induce inflammation. And so we'll take a look at what happens during inflammation right now. So everybody probably knows what inflammation looks like. So here's someone that has an inflamed left eye. And so if you were to describe, like if you were to say, well, how do you know that inflammation is happening? Well, here are some signs that are associated with inflammation. So if you have redness, swelling, heat, pain, and loss of function, those are all things that characterize inflammation. And basically the reason why all of this happens during inflammation is because all inflammation is the rush of blood fluids to a particular area. And there are two things that can cause blood fluids to rush to a particular area. One of the things is infection with a microorganism. Another thing is tissue damage. And the reason why, again, it's useful to have blood fluids rush to an area if it's infected, for example, is because these blood fluids contain a bunch of different things that can fight off that infection. So, for example, some stuff that you'll find in these blood fluids include neutrophils, macrophages, complement proteins, antibodies, and also clotting factors to help the wound heal if it's tissue damage that's causing the inflammation. So what we'll do next is say a little bit more about how infection with bacteria can cause inflammation to happen. So we'll use this little diagram here to show you that. So to break this down, so there's quite a few things that's being shown in this diagram. So let's break it down to explain what's being shown to you. So here is the surface of the body somewhere. This is showing that a wound has happened, and associated with that wound, bacteria have now infiltrated the body. What you have here deep in the body is a blood vessel, and blood vessels contain blood, which has red blood cells in it, and certain types of white blood cells, for example, neutrophils. Out here in the tissue, you find different types of white blood cells too. So you find mast cells, which probably should not be a surprise, right? Because we've already mentioned that mast cells play a big role in inflammation. There's also platelets that have found their way out into the tissue, and what they're going to try to do is to close off that wound. And also in the tissue are macrophages, right? So then here are all the players that are involved when inflammation occurs in response to an infection. So if we kind of go step by step as far as what happens, here's what you find. So it turns out that mast cells contain molecules on their surface that allow them to bind to bacteria. So let's say that happens. So a wound occurs, bacteria are now infiltrating the body. There's a really good chance that one of them will bind to molecules that you find on the mast cell. What you might remember mast cells doing is that they are able to release histamine. And so this is one of the things that would cause mast cells to release histamine, is if a bacterial cell binds to certain molecules on its surface, causes it to release its payload, its histamine. And what histamine does, so before we mentioned that histamine is a molecule that induces inflammation. So more specifically, what histamine does is that it travels to, or it binds to blood vessel cells. So the cells that line the blood vessels actually have receptors on them that can attach to histamine molecules. And when that binding happens, it signals these cells that line the blood vessels to loosen their interaction with one another so that they become more spread apart. So let's jot that down here. So histamine causes the cells that line the blood vessels to space further apart. And the reason for doing that is because it creates an exit, right, for the white blood cells in the blood vessels to be able to leave the blood vessels towards this infected area. Let's mention this. So there's actually things that then induce those white blood cells to leave the exit once those exits become available. And so here's how that happens. We mentioned that another type of cell that you find in the body's tissues are these macrophages. We've already mentioned that they have things like toll-like receptors on their surface that are able to bind to things that you find on bacteria. It turns out that one of the things that macrophages do in response to binding to bacteria is that they release cytokines. Specifically, there are macrophages that release a type of cytokine called a chemokine. And so you might remember that what chemokines do is that they cause chemotaxis of white blood cells. In other words, they attract white blood cells into a particular area. And what this is showing, what this picture is showing, is that specifically what this macrophage is doing is that it's releasing cytokines that end up attracting neutrophils into the infected area. So let's mention that here. So macrophages, after a microbe binds to a pattern recognition receptor, like a toll-like receptor on that cell, that macrophage then ends up releasing cytokines that attract neutrophils from the blood vessel out into the tissue. And again, just to reinforce some of the terms that we've used, whenever you're talking about a cytokine that specifically acts to attract other white blood cells into an area, that's a type of cytokine called a chemokine. Once all of these reinforcements, once all of these neutrophils, for example, have gotten called into this infected area, now we're in business. Now it's game on, because these neutrophils can do things like phagocytize the bacteria. These neutrophils can also release toxic substances. They can release antimicrobials that can kill off that bacteria. So now we're in the process then of resolving or getting rid of all of these bacteria. It turns out that these neutrophils have a relatively short lifespan though. So after they release their granules, for example, that contains antimicrobial substances to kill the bacteria, the neutrophils end up dying soon after that. And so you can actually see the collection of dead neutrophils that end up forming, because that's what pus is. So all of that whitish yellow fluid that is associated with inflammation, all that is dead neutrophils for the most part. Right, so again, those neutrophils die after shortly after releasing their granules, and that's what pus is. It's mostly dead neutrophils. And eventually all that pus, all the dead neutrophils end up being cleared by macrophages that phagocytose them, chop them up even smaller, and then get rid of them after that. Right, so then that's how an infection, so that's how infiltration of bacteria can induce inflammation. And this is the reason why inflammation is useful during an infection, because it brings on a whole bunch of different types of helpful things like neutrophils that can get rid of those bacteria. It turns out that bacteria aren't the only thing that can induce inflammation. Tissue damage can also induce inflammation. And so basically it's the same process, except what's triggering it now is not necessarily bacteria, but damage to our own cells. And so it turns out that whenever our cells are damaged, some of the debris that ends up being released can end up activating mast cells and macrophages to do what we just explained, right? So it can induce inflammation in that way. So let's just mention that here as the take-home point, that tissue damage, again, can also induce inflammation, kind of like bacterial infection can induce inflammation. The reason why tissue damage can induce inflammation is because the debris that ends up being released as a result of that tissue damage can bind to mast cells and can bind to macrophages in order to cause those cells to become activated. All right, so if we take a step back again to look at the big picture, again, we're taking a look at our innate immune defenses against microorganisms. We mentioned that you have these cells in the blood that are considered part of the innate defenses. Those cells basically do three things: phagocytosis, inflammation, and fever. We've already talked about what happens during phagocytosis. We've just finished talking about what induces inflammation. And so what we'll do last here is take a look at fever and see how it is induced. So again, when you're talking about fever, what fever is, it's a raise in it's an increase in body temperature as a defense against infection by pathogens. And basically the reason why fever is useful is because of two things. So one of the things that fever does is that it just takes pathogens out of their optimal growth range, right? So if you have bacteria, for example, that are optimized to grow in the human body, that means that they're optimized to grow at 37 degrees Celsius. So if you change that temperature a little bit, then it would just take the bacteria, the pathogen, out of their optimal growth range, so they would not grow as well. So one of the ways that fever ends up fighting pathogens is, again, it just takes the pathogens out of their comfort zone, so it eliminates their optimal growth temperature. Another thing that fever does, another thing that a raise in the temperature does, is that it increases the activity of enzymes that are involved in fighting off the pathogens. So for example, it might increase the activity of the lysozymes that are found in lysosomes and other enzymes as well. So fever is useful if there's an infection, basically for these two reasons. If you're considering what directly causes fever to occur, it's a type of molecule called a pyrogen, right? So again, pyrogen is a substance that induces fever. It turns out that there are different things that produce and release pyrogens. Like you might guess, foreign things like bacteria have molecules that are considered pyrogens because they induce fever. And if you're talking about that specific type of pyrogen, one that's made by foreign cells like bacteria, those are called exogenous pyrogens. And if you break down this term exogenous, so exo means outside, right? So these are pyrogens that are made by things outside of our own cells. Specifically, they're made by foreign cells like bacteria. It turns out that pyrogens, in other words, fever-inducing molecules, can also be produced by our own cells. And so when you're talking about those types of pyrogens made inside of our own cells, those are called endogenous pyrogen. So endo, that prefix refers to inside, again, because these are pyrogens or fever-inducing molecules that are made inside of our own cells. Both of these types of pyrogens, so pyrogens in general, what they do to increase the body temperature is that they travel up to the hypothalamus in the brain, and they cause a change up there. And so it's the hypothalamus that ends up regulating the body temperature. And so any change of activity in the hypothalamus region of the brain would then change the body's temperature. And so pyrogens interact with the hypothalamus region of the brain. And so to make sense of all of this, it might help to use a picture to describe what's going on. So in this picture, you're being shown someone's brain, the person's liver, and then here's a bunch of cells that are associated with immune defense that are found in that person's body. What we just mentioned is that there is a region in the brain called the hypothalamus that's responsible for regulating body temperature. If there happen to be any pyrogens that interact with the hypothalamus, that is what causes an increase in body temperature. In other words, that's what causes fever. We mentioned that there are two general types of pyrogens. There are these things called endogenous pyrogens and these things called exogenous pyrogens. Endogenous pyrogens are fever-inducing molecules that are produced by our own cells. So this is showing you that macrophages found in the liver can release endogenous pyrogens. And so Kupffer cell just refers to a macrophage that's found in the liver. This picture is also showing that there are types of white blood cells called monocytes that can release fever-inducing molecules. In other words, they can release these endogenous pyrogens that tell the hypothalamus to increase the body temperature. You might be looking at this term monocyte and thinking, wait a minute, you didn't have this cell monocyte listed in your diagram that described the many types of white blood cells that are out there. So we'll mention it now. Basically, a monocyte, it's a cell that ends up giving rise to the macrophages that you find in the blood. Right? So this is the mother cell of macrophages that end up in the tissue. And then also, we mentioned that there are these exogenous pyrogens. So all of these little dots here represent bacteria. They also have substances. And actually, the lipid A portion of lipopolysaccharide that you find in some bacteria, that lipid A actually induces fever. So if any of that is released and finds its way up into the hypothalamus of the brain, it causes an increase in body temperature. So lipid A is an example of a pyrogen that is produced by bacteria. In other words, lipid A is an example of exogenous antigen. So again, there are things in bacteria that cause us to produce fever, which is a good thing for us, that we respond by making fever, because then it acts to slow down the bacteria. Whenever some of our own cells, like macrophages, come into contact with bacteria, they also are able to release cytokines that end up causing fever. Again, that's a good thing because it then will end up slowing down that bacterial infection. So that's what then ends up causing fever, and that's why fever is useful. And that's basically the story on innate immunity, right? So these, again, are all the components of the innate immune response. These are the things that the cells in the blood that are considered part of the innate immune response. These are different things that they can do to slow down infection. What we'll do in class, so in this video we haven't really talked much about complement proteins, so we'll save that for class. So we'll save that, we'll do an in-class lecture on what complement proteins can do. And then starting next week, so to take a look at the big picture, right, what we've taken a look at this video, or what we took a look at in this video, are the innate defenses. So all in next week then, we're going to just focus in on the adaptive immune defenses and see what all they can do.